Hello, I am Hudi Bahar Kasseh, and this is my science fair project, the molecular docking analysis of the receptor binding domain of SARS-CoV-2. All nations around the globe have been shocked by the SARS-CoV-2, which has started to spread from China to all countries around the world. The SARS-CoV-2 has a positive sense single-strand RNA genome and belongs to the beta coronavirus familia. It is compared to the HIV virus in structure. The virus has infected millions of people and has killed hundreds of thousands of others. Yet the humankind has not been able to 100% stop the spread of the virus. How does this virus come into our body? The SARS-CoV-2 utilizes the receptor binding domain on the spike protein to bind to the ACE2, also known as the angiotensin converting enzyme 2, on host cells and enters the cell through the endosomal pathway 3. Once inside the cell, RNA is released and serves as a template for viral proteins and for making copies of the genome. Here's what the spike protein looks like in relative to the rest of the coronavirus genome. The spike protein has two parts, S1 and S2. We are more focused on S1 because that is where the receptor binding domain lies. Here's an image of how the novel coronavirus binds into the human cell. This is where the spike protein is. It attaches itself to the ACE2 and infects the rest of the human body. Here's what the receptor binding domain looks like in relative to the rest of the spike protein. As you can see, it is located at the very tip of the S1 unit. So what is the problem? The COVID-19 virus uses the receptor binding domain on S1 in order to attach itself to the host cell and starts the infection. We need to block the entry of the receptor binding domain to the organism's cells. What is the purpose of my project? The purpose of my project is to find a way to block the receptor binding domain protein of the coronavirus from entering into the living organism's cell. Wouldn't it make sense to find chemicals and medicine to block this entrance so that we can prevent it from infecting the rest of the human body? Can we find a strong enough ligand using the virtual screening method to block the receptor binding protein part of the coronavirus from entering the host cell? My prediction is that I will be able to find at least some ligands to block the RBD from attaching the virus to its host cell using virtual screening. Here's the materials that I used. Autodoc MGL tool, Autodoc Vina, PyMol, OpenBabel, the BASH program, our target protein, and ligands to test. These all were downloaded onto my computer. I chose certain ligands because they were proven to be strong against viruses with similar structure. Therefore, when I researched for ligands, I typed in ligands strong against alpha and beta coronaviruses. First, we need to download the protein. So we go into Protein Data Bank and find our protein. Mine was 6M17. I downloaded into my file in PDF format and then dragged it into Autodoc tool. This is where I have to convert it into PDB-QT format. But before I do that, I have to simplify my protein so that my ligand can easily find where to bind to and block the RBD or other parts of the protein. In order to do that, first I deleted water, then I added a polar hydrogen and a Coleman charge, then set my protein up into 84 format, and then removed the head atoms. Now my protein is ready to be downloaded in its final form through grid. Here's what my protein looked like after it was simplified. I chose to do 6M17E rather than 6M17F. Next, we have to determine the binding site. In order to find the binding site, first I had to read published papers on PubChem. Then I went on Autodoc Vina and clicked Grade. Then I arranged the size and the center so that I can create blocks where the ligand can bind to and easily stop the receptor binding domain. Then I made a coded configuration file based off of the data that I found. Here's what my configuration file looked like. Next, I'm ready to prepare my ligands. First, I go into PubChem and search for my ligands. Then I download them in SDF format. Here are 126 ligands downloaded into my files in SDF format. Afterwards, I have to convert my ligands into PDB-QT. This is what I used in order to code for that. Afterwards, I label all of them with an L at the beginning so that the program I created can easily read my ligands. Here's what the run1 file looks like in order to read the ligands. Next, I'm ready to finally run the data on all of my ligands. To run them all at the same time, the following files are needed. The ligands, our run1 file, a command prompt, and a configuration file. Here's what this looked like. I opened my command prompt, typed in CD, and dragged in my file, and clicked enter. This brings us inside of the file. Then I typed in bash space run1.sh. Now my computer is running the data for each of my ligands. After that is done, all of the data for each of the ligands are saved into separate file folders per ligand. Here's what that looks like. And finally, my results. I tested a total of 126 ligands since the end of October and used one batch file and checked their binding energy level to create a spreadsheet to check how strongly they blocked the receptor binding domain from binding to the host. I analyzed my results according to the criteria following. Any binding energy lower than negative 8 kcal per molecule should be considered as a strong candidate. Refer to the following result tables. My research results have showed that my hypothesis was correct. Structure-based virtual screening helped me find ligands, block the binding site, and prevented the COVID-19 from growing into the host. I was able to find quite a number of ligands with the binding energy level lower than negative 8 cal per molecule. This means that I may be able to create a medicine using the successful ligands and potentially find a substitute vaccine to stop the SARS-CoV-2. There were 17 results out of 126 ligands tested that were below negative 8 kcal per molecule. It was less than I expected, but it gives enough information for our future research. The best of the best was exocarinin at negative 18 for a binding energy level. The lowest of the best was tetradyne with negative 8.1 binding energy level. Future research. As a follow-up for this project, I am planning on experimenting this computerized research in a real-life laboratory. There are a few parts missing in this first part of the project. Firstly, we don't know which of the ligands may be toxic for an organism. We also don't know what combination of ligands are toxic for the organism. We plan on creating these and testing them in a safe environment. Thank you for listening to my project.